Good morning, everyone. Thank you, Malo, for the kind introduction. And yeah, so this morning I wanted to talk to you about two main projects I'm currently conducting in the lab of Kenneth Birnbaum here at New York University. And so the first one is, I will be mostly talking about root regeneration, which has been kind of the main core research in the lab for the past few years, and how we use a single cell RNA-seq in Arabidopsis to better understand what's going on there. And so in the second part of the talk, we will move from Arabidopsis to other plants, such as crops, and how we use single cell and single nuclei RNA-seq to identify novel marker genes in non-model species, and how we can use these different data set to actually do pretty cool analysis of gene conservation and transcriptome comparison across species. So as you all know, because we are plants lab, right? Plants are the real master in regeneration. And I'm pretty sure that many of you have already tried that. You take your favorite plant, you cut it, you stick it into water, and you get roots out of stem. And so in a biological point of view, it's very interesting to see that cells that are already maturated, already doing their jobs here as a stem, if you change the environment, these cells are able to fully change our identity coming from a stem to a root. And so that's a little bit what we're interested in the lab, and how is it happening, and what are the major molecular mechanisms that allow this transition of cell and changing a cell fate. So to do that, we use the model plant Arabidopsis thaliana, and here is a representation of the root meristem we are very interested in. Because, so you all know pretty much this root meristem, it's extremely well structured and patterned. And at the very center of it, you have the stem cell niche, which constitutively divide throughout the life of the plant, giving rise to all the different cell files here, making the mature roots. And what's very interesting about this model system is that if you cut the root, if you cut and you remove the entire stem cell niche, the root is still able to fully regenerate in about 2 to 3 days. And so what's pretty crazy is that here cells that are already identified as cortex or stele or endodermis then are able to change our identity, restart a division, and reform a functional meristem. And so when you look at different marker genes, here's an example with WOX5 GFP reporter that is only expressed in the stem cells, usually. You cut that away, and you see this reporter coming up after 1 day in other cells here above the wounding site. And so by day 2, you have like these markers, it is focusing in the center. And then by day 3, you have a novel stem cell arising. So what's pretty cool about it is that there is no cell movement. It only relies on cell division, novel cells, and so cell transition. There are many people having their microphone on. If they can mute themselves.
Pierre-Marc, ton micro.
Sorry about that. No problem. So to better understand what's happening there, we decided to go for a single cell approach where we took all the different time points throughout the regeneration process and also uncut conditions, and we did single cell RNA-seq. So for people who are not really familiar about this technique, I just wanted to go very fast on it. And what we do basically is we take plants, here Arabidopsis growing in vitro, we take the root we're interested and we digest it using a specific cocktail of enzyme that's going to digest the cell wall and release the cells into a solution that we call protoplast. And so what we do after that is we use a system developed by 10x Genomics, which is using this machine that is actually not bigger than a bread toaster. It's like a small machine and it's very complicated. You have one button to push here. So it is rather straightforward. And so the technique relies on, you put here in the chip, here, you have chip with different wells. And so in one well, you put cells, in another well, you put oil, and in a third well, you put beads here that are barcoded beads. So each bead has a unique barcode and are covered with poly-T that are going to trap messenger RNA. So what the machine does is that it's pushing these three components through a microfluidic system. And here you see that actually the beads and the cells are combined into droplets of oil. And so as an output, you get thousands of droplets containing a bead and a cell. So the bead is going to catch the messenger RNA, and then cDNA is synthesized into each droplet. So using this technique, we are able to sequence about between 4,000 and 6,000 cells per replicates in a matter of very few. So to go through the machine, it takes about 9 minutes for these 6,000 cells to go. So it's rather fast. And so what we do after, when we sequence the data set, we include everything using an R package called Seurat. And so this R package is going to cluster the cells together according to their transcriptome without us giving it prior knowledge. So it's a blind clustering. And so it gives you this kind of maps that we actually call UMaps, where each dot here is a sequence cell. And so the program clusters them together according to the transcriptome. And then the big game is to identify which cells is which. So using different reporter genes. So here in our data set, we were able to identify all the different components of the uncut root, and here labeled with the same colors as the skim. So we were able to find columella, stele, epidermis, stem cells, and so on. And when we look at the cut cells this time, we were surprised to see actually quite the same thing. And it's not, when you think about it, it's not that surprising, because when you look at the roots that are in regeneration, we are only interested into these cells here at the wounding site that are regenerating and changing your identity. But all the cells here above, even if we try to cut or enrich for the wounding site, are actually normal cells, uncut cells that we find here. So Seurat wasn't doing a great job at identifying these cells that are in regeneration. So we need another approach to refine or to identify like a very specific signal of these cells. So to do so, we went for topic modeling approach. And so I'm going to summarize that very quickly. I hope it will be easy to understand, but basically you start with an expression matrix where all your cells are in column, and then you have all the genes. And the topic modeling is an algorithm that actually will identify co-expressed genes and will summarize them as topic. So under a topic, you have a few dozen, sometimes 100 genes that are co-expressed in a group of cells. And so you can summarize then the transcriptome of a cell by a combination of topic at certain frequencies. And so typically it gives you graphs like that. I don't know if you ever seen such things, where each cell is summarized by kind of a colorful barcode, combining different topics at different proportion. And so you can now identify different cells identity, and you see this identity 1 is characterized by a high proportion of this brown topic, while identity 2 is mostly about the purple topic, and so on. So we perform this topic modeling in our data set, first in the uncut data. And here are the results, where we can see very specific differences between cell type, where the columella lateral root cap is mostly characterized by the yellow topic, as you can see here, while the stele is mostly characterized by a purple topic, for example. And so we can trace back what are the genes. You don't pick the genes, the program picks them for you, but then you trace what are the genes underlying this topic. And so when we look at the cut data sets, actually what we found is that in the cut, we see the rise of a new topic that is only expressed in the cut dataset and not in the uncut. And this is the orange topic. So you can see here that actually here, lots of cells are getting this topic. And when you look at the same cluster, the young columella, this topic is not present in the uncut conditions. So we thought this is a good, this is looking good, and we identify that under this orange topic, there were lots of genes already known, important for root morphogenesis and seed development. And so now we have like a multiple candidate genes that seems to participate to root regeneration. So then we were wondering, where are these cells located in our map? Are they all located a bit randomly, or do they have a coherent behavior? So when we map these cells that are actually the cell that we consider now in regeneration in our data set, here is the exact same UMAP I showed you in the past, and I just label the cells in regeneration in orange in the different time points. So at 4 hours, we found that these cells were actually located in the stem cells or endocortex identity, and a few already having like a columella lateral root cap identity. And by 9 hours or even 14 hours, we see an enrichment of these cells that are converging towards the lateral root cap columella. And sees the same in 24 hours. And by 36 hours, we start to lose the signal, because by 36, the root is mostly regenerated. And so very few cells need to change our identity, and you kind of go back to the original stage. So using this approach, we're able to identify cells in regeneration at all the different time points. And we were able to build trajectories and identify differentially expressed genes across this data set. And so especially we were very interested because we identify about 100 transcription factors that seems to be extremely important in regeneration. And so we are currently screening for these candidate genes using a bunch of CRISPR lines or a newly published inducible CRISPR, which is working extremely great. We are also we're getting the help of a bioinformatician we recently hired in the lab that is working on machine learning models to confirm this signal of cell in regeneration and maybe extend it and make it more robust. So it's a work in progress to use machine learning for that. But so we were very happy. We had a quite understanding of what's happening, but then I wanted to talk to you a bit about mechanism and how is it possible that a change, that the cell, is able to change? What has the cells need to do to move from, or to change its identity from here, like a cortex cell to a new columella cells? And so in the field of regeneration, the main question that's been going on through years is actually, can you transdifferentiate? Can you jump from one identity to another? Or do you have to go through a stem cell-like state? So that's what we kind of started as a question. And what are the mechanisms important to change directly or not? So we went for two very simple models first. We went for a direct inhibition model, where first you need to lose your old identity before a new one can arise. And so there is like an inhibition of the old model preventing the new one. Or we have the passive loss model, which is you lose your old identity progressively. Meantime, you also have a new identity that's rising. So to test these different models, we looked at our data sets. And here's an example when we look at the still cells here in the center in pink. And we combine all the marker genes we have identified that define what still cells are, and we give them a score. So in the uncut condition, the still cells have a maximum score of 1. And then we look at this score during regeneration in the cells in regeneration. And so what we find is that as soon as 4 hours, these cells start to lose the cell identity. And by 14 or 24 hours, they're no longer still. But when we look at the same cells, what new identity is rising there? We found a lot of columella markers were rising as early also as 4 hours, really pointing towards the second model of the passive loss and overlapping. But mechanistically speaking, we weren't sure what was happening. And we were wondering, what if we try to block this loss of old identity? What if we artificially maintain this old identity and force it to remain? And so the hypothesis was that you lose very fast your identity by closing the chromatin. And so we investigate what if we avoid this chromatin closure, and is it going to work the same way? So to do that, we investigated the use of trichostatin A, which is an inhibitor of histone deacetylase, and basically a block chromatin closure. So we first did a very simple test using our regeneration assay, where basically we cut the root and then we let the root regenerate, and about 2 days, you get a full new meristem. So here's the control with TMSO, where we have 100% of regeneration. But when we treat with TSA, as early as 1 hour post-cut, and then we put back on regular medium, we found that TSA greatly impairs the regeneration efficiency. But what was even more cool, I think, is that if you cut on normal media and then you treat with TSA, so here we cut, we've waited 4 hours, and we treated for 1 hour with TSA and put back in regular medium, then we don't have any effect of TSA. So it seems that TSA effect or role is extremely important at the beginning, and maybe shutting down multiple genes, maybe stress-related genes, to allow a good regeneration. So to have better understanding of what was going on, we went for single-cell RNA-seq. And what we did is we treated the root with TSA for 14 hours, so kind of a harsh treatment to be sure we have a significant transcriptional response. And we looked at marker genes that we knew from the previous analysis. And when we look at marker genes here at 14 hours, we can see that when we treat with TSA, we indeed retain kind of an old identity to remain. And so by 14 hours, you get like a 50% still identity remaining compared to almost nothing in the control conditions. And then we looked, okay, but are these cells capable of getting new identity, or are they just stop? And when we look at that, we actually were very surprised to see that cells don't really care and still get new columnar identity at a lesser rate than the control, but still at a significant rate. So it means that even though TFA is blocking regeneration, it doesn't block the rise of a new one, not fully at least. And so now we're working on multiple candidate genes and transcription factors, and we characterize them. So we characterize this transcription factor as the early ones that are expressed early on during the regeneration process, the middle ones that are expressed from the beginning to the very end, and the late ones that are only expressed later on. And so now we're characterizing which are affected by TSA. And you can see that, for example, our favorite WOX5 is not affected by TSA, Bravo, or TMO6, while FEZ is greatly affected. And among all these transcription factors, only 22% were perturbed, which was not actually as many as we thought. And some other, like Monopterus, they don't care, they just still express fine with TSA. However, Baby Boom or Plethora2, which are two crucial genes, are actually not expressing TSA. So we are characterizing these different behavior. And we also, of course, validate the single-cell RNA-seq by using a reporter genes, or reporter lines, sorry. And here is WOX5 expression during regeneration, so cut 1 day after, and you see its expression here. And when with TSA, even if it's expressed at a lesser rate, it's still expressed for WOX5. Same for WORKY23, it's expressed in TSA, but other genes that we found in single cell, such as WIP4, FEZ, or Sombrero, don't come back when you treat with TSA. So from that, we kind of ended up to have like a mixed model from the question we raised. And we think that this loss of old identity is partially mediated by chromatin closing. And at the same time, it still allows a new identity to arise. However, you need to lose enough old identity. And there is like a kind of a partial repression of the old identity to the new one. and you need to lose enough to be able to fully differentiate and get a new identity. And so what we do with TSA is that we block this loss, and we end up to have like a partial dual stage where there is no new identity, and so there is no, not fully new identity and no regeneration. So that's a little part of this project, because we're also working on cell cycle. And I'm not going to talk about it because I don't have that much time, but we also wonder, do you need to divide to fully change your identity, or can you, how far can you change without division? So that's, we're using cell cycle inhibitors and combined with single cell RNAseq. And similarly, do you need chromatin opening to get new identity? So we're working with inhibitors of this pathway as well. And so if you're interested, like, feel free to ask me any questions later on about that. Okay, so the second project I wanted to talk to you about is the use of a single cell and single nuclei RNA-seq to identify new markers in non-model species, and also have kind of a direct comparison of transcriptome to map cellular divergence in crop plants. And so it all started a few months ago, where a postdoc, Carlos Ortiz Ramirez, in our lab, started to characterize for the first time the root meristem of maize. And so he was able to successfully do the protoplasting and the single cell and identify almost every cell types present in the root here. And he was able to create like a very nice story about the scarecrow and short root interaction in maize that is actually, we thought it was very stable across, short root and scarecrow interaction is extremely well studied in Arabidopsis. And actually we found that this interaction is extremely different in maize. And so these genes are not expressed in the same cell type and everything, so yeah. So we were actually very happy because the manuscript just got accepted in Science last week. So you will be able to see it, I think, mid-December if you have further question about it. But so one major drawback of this study is that no matter how hard he tried to find every cell type, he couldn't find any columella cells in his data set. And columella here, if you see the diagram, is quite a big part of the roots. And it's kind of a shame to not have it. And if in fact the problem is that the cell wall in columella is extremely tough. And so using the enzyme we tried, we cannot get a good cell from it because we cannot digest it. And it's a general case, like a general issue for many plants. When you move out from Arabidopsis, the cell wall is much more tough. And especially when you're interested in more mature organ, it's almost impossible to get good cells. So we developed a single nuclei RNA-seq approach to be able to overcome this issue. So we just take the root, grind it, extract the nuclei, and do the exact same thing as single cell. And so when we mapped our single cell and single nuclei in maize meristems, you see here the same UMAP with cells in blue and the nuclei in red. And at the very center of this UMAP, you see a kind of an island which is only composed of nuclei individuals, while all the other cell types are kind of mixed. And when we look at the marker genes we know from this publication, we could identify that these cells are actually, these nuclei are actually the columella. So we're quite happy because by combining single cell and single nuclei, then we are able to have a better representation of all the different cell types present in the root meristem. And so we wanted to see, okay, can we use our knowledge in maize to look at other species at a single cell level? And it's extremely important right now in a global warming to find like other species that are also much more adaptive to stresses than maize and more heat and drought tolerant. And so usually these species are much less studied than maize. So we went for a very kind of expected candidate, which is sorghum bicolor. Because sorghum and maize have diverged only a few million years ago, about 10 to 12 million years ago, and they're quite similar genetically speaking. And then we thought to pick an outgroup, which is a kind of a new rising model plant in many labs called Ceteria viridis, and it's kind of the wild type of the millet. And so we thought, can we use what we know about maize to jump from one species to another and use our data set here to map the other data sets? So we performed single cell and single nuclei RNA-seq on the roots, the primary roots of these three species, and look at the autologous genes in between. So among all the genes we detected for all of the different cell types or different replicates, we were able to identify 41% of these genes were direct autologs in maize, and 36 were direct autolog in maize as well. And so we use a previously very well-made studies where they were looking at autologs across multiple grass species. And so we wanted to see if we were able to use these autologous genes to map Ceteria and sorghum dataset into the maize one. And so we used the same R package Seurat to do so. And Seurat was extremely efficient at assigning cell identity or assigning cells to the maize dataset we had. And so using the maize markers, we're able to identify all the different cell types here in our single cell and single nuclei dataset with epidermis, columella, all the cortical cells, pericycle, stele, endodermis, and so on. So, of course, we were very skeptical at first. Is it that true? Is it working that well? And so we collaborated with the Gillis lab here in Cold Spring Harbor, who are specialized in several species comparison of transcriptome. And so they conducted several independent analyses, such as meta-neighbor analysis, which is basically you take each cells blindly, you don't know to which identity it's assigned, and based on its transcriptome, the algorithm tries to predict its assignment. And depending on how well it predicts, you get a confidence score and then a lot of statistical validation of that your cell is well attributed. And but we are currently validating all these different cell type by a bunch of in situ hybridization in the 3 different species. And because I never did in situs before, and I've tried, and it's such a pain to just embed, do the slicing and everything, and I'm pretty lazy, I have to admit. So instead of doing that, we developed a whole mount in situ hybridization in maize, sorghum, and Ceteria using fluorescent probes. And so here's an example of a positive control where it's GRP4 in maize that is already published a few years ago and was characterized to be expressed here in the epidermis and lateral root cap columella. And when we look at our whole mount in situ hybridization, we could confirm that its expression was indeed in the lateral root cap that was previously published, and also actually in the 2 first layers of the epidermis in maize. So now we're confirming with its autologs in sorghum and in Ceteria. And because using this map, now we have like dozens of marker genes for all 3 species. And so we are just currently validating these. And here's another example of autolog genes between maize, Ceteria, its autologs in Ceteria and autolog in sorghum that are all 3 extremely well localized in their expression in the endodermis. And so this is one of the candidate genes we are actually testing across. And here's another in situ hybridization whole mount as well of another positive control, a scarecrow 1 in maize. And we could confirm like it's already published very neat expression in the endodermis. So if you're interested in this protocol, we can further discuss about it later on. So now we are very happy because we had an understandable map of different cell type across 3 species, and now we can have fun. Because the cool thing about that is that you can study how gene evolve and how transcriptome evolve across species. And what's pretty cool is that upon the divergence between maize and sorghum, actually maize went into a whole genome duplication event. And so if you had a gene A in maize, it's been fully duplicated into two subgenomes, maize1 and maize2. And then throughout evolution, these maize1 or maize2 copies were either selected or lost. And so in the modern maize, sometimes you find only the maize1 homologs was retained, sometimes only the maize2 and the maize1 was lost. But in certain cases, you have maize1 and maize2 homologs that were conserved. And so it's still not really understood in science, in plants development, how plants are actually co-opting with that. And what is the expression pattern of these two homologs? Are they expressed in the same cell type? Is maize1 more specialized in certain cells than maize2? These kind of things. So we use our data set to try to understand that and compare how maize1 and maize2 homologs were expressed compared to their sorghum or Ceteria ancestral gene. So we first went for a very simple approach of co-expression. Are these maize1 and maize2 co-expressed with sorghum? And so here is a general co-expression of all the different cell types. And so here you see that 46% of these genes are actually co-expressed in the same cell type between sorghum, maize1, and maize2. However, you have other genes, other orthologs, where they only expressed in sorghum maize1, and the maize2 orthologs is not expressed in this cell type. And you have other genes that are only expressed, for example, in sorghum. And now the maize1 and maize2 orthologs somehow are not expressed in the same cell type. So when we look at geoterms associated with these different categories, we found that these co-expressed genes, 46%, were highly enriched for cell homeostasis and general cells everyday life, let's say, with ATP metabolism, translation, ribosome biosynthesis, and so on. However, in the other categories, we found that these different genes were actually enriched for adaptive responses and adaptation in general, because we could find a response to toxic substance, detection of stimulus, and defense and immune responses. So it's very interesting because this gene would be a primary candidate to see what's the difference between maize and sorghum, and maybe these 16% are genes that are very specialized in sorghum, and maybe that makes sorghum more resistant to drought or heat or certain pathogens. So this could be of primary interest. To go a bit further into the evolution point of view, we look at the KAKS ratio of this different gene category. So KAKS is a score that you assign about the substitution rate of this gene. And so basically, if a gene is extremely important and conserved, then the substitution rate is low because evolution tends to force this sequence to be maintained during time. However, if a gene is less important or under less selection pressure, its KAKS ratio will be higher. And so what we found is when we compare the KAKS for all the different gene categories, we found that the co-express orthologs, the sorghum, maize1, and maize2, actually have a significantly lower KAKS ratio, confirming they are under a stronger selective pressure than the other genes. And another cool analysis we found is that when you look at genes that are only expressed in sorghum and maize1, and the maize2 orthologs is not expressed there, we find that this maize2 that is not expressed has a much higher, significantly higher KAKS ratio, showing that S and maize1 are under selection pressure, but maize2 is less. And so these are the first glimpse maybe of a new functionalization or more a first drifting of maize2 in this context. And I will show you a few examples in 2 slides. Okay, so starting from that, we then ask another question that's a very genuine question. But basically, if you're a cell, okay, and you have duplicated gene, MEIS1 and MEIS2, express in the cell at the same time, are these expressions similar? Meaning that if you have 2 copies of the gene, are these 2 copies expressed at the same level, and therefore you get twice the amount of protein you would get with 1 copy? Or is there a compensation mechanism? And that's not really understood for homologous genes like that. So to test this hypothesis, we compare the expression of MEIS1, MEIS2 to its sorghum autologs in every cell types. And so we did so by just doing the ratio of sorghum expression over maize. And here's the distribution. When you look at genes that are only expressed between sorghum and MEIS1 in one cell type and MEIS2 is not expressed, the ratio of expression is actually the distribution in center towards 1, which means that they have roughly the same expression level. It's similar if you look at genes that are only expressed in sorghum and MEIS2. However, when you look at genes that are co-expressed with sorghum, MEIS1, and MEIS2 in the same cell type, then the ratio are much different. And you see it's quite higher, so meaning that the sorghum autologs is highly expressed compared to the MEIS1 or more highly expressed compared to MEIS2. And so to get to the level of sorghum expression, you need to combine the expression of MEIS1 and MEIS2 to recenter the distribution towards 1. So here, using our approach, we're able to kind of prove these compensation mechanisms when a cell has double the amount of gene that seems to be still expressed together, but at a lower level compared to the unique copy in another species. And so what's pretty cool is that you don't see that very clearly if you do a bulk RNA-seq, taking all the cell type together, because here it's really cell type per cell type dependent. And so starting from that, we then looked at candidate genes that could be interesting to understand why sorghum is so heat and drought tolerant. And so our choice went for a very obvious reason to aquaporins, because aquaporins are extensively studied in this species as a primary genes for drought resistance. And we were very lucky because we found an aquaporin type 2.-2 that actually had this MEIS1, MEIS2 present in the maize genome. So here's a dot plot representing a type 2-2 expression across the different subgenomes. Here you have the different cell types, and here are the dots. The bigger the dot is, the more cells expressing the gene, and the level of the color it represents the expression levels. And so when you look at their expression, you see that in cortical cells, in all the different cortex cells, sorghum is co-expressed with the MEIS1 and the MEIS2 autologs. MEIS1 is always a bit more expressed than MEIS2, but they still co-express. However, what's pretty nice is that when you look in trichoblast, then it's only sorghum and MEIS2 that are co-localized or co-expressed. But when you look at still, and actually also pericycle xylem, it's only the MEIS1 autologs that is co-expressed with sorghum. So here is a first example of a neo-functionalization or a sub-functionalization where MEIS2 is mostly specialized in trichoblast expression, while MEIS1 is mostly specialized in still expression. So I thought it was pretty cool to show that. And these are this kind of genes we're looking for to better understand what's going on there. And so we have several other aquaporins we're interested. Some here, PIP1-3 is actually extremely well conserved across species in sorghum, maize, and Ceteria. But other aquaporins, such as NIP1, are more specialized in sorghum. And you see here that NIP1 is only expressed in the sorghum endodermis, but not in maize or Ceteria, similar for still or pericycle. So we are trying to do some functionalization using already present, using different library lines and insertion lines. So one last analysis, and then I will close this presentation, was, okay, we have now access to the transcriptome of every cell type in 3 different species. And our question was, are these transcriptomes similar between species, or is there some cells that seems to have a more similar transcriptome across species and some cells that seems to be more divergent or not? So also in collaboration with Gilly's lab at Cold Spring Harbor and combining an approach that they were using called CocoConnect, we, we assigned a score of conservation to every cell type transcriptomes. So here is a score of conservation with Ceteria. And so we look at maize and sorghum transcriptome and try to see, using autologous genes, which transcriptome was more similar to Ceteria and which one was the more divergent. And what we found is that actually that the epidermis and the initial cells were much more conserved than other cell types. And when you look, for example, at cortical cells here, cortex 2 and 3, or columella, these transcriptomes seems to be much more divergent across species. So here, for the first time, we have access to the information to show that there is kind of a differential evolutionary rate at the transcription level between cell type, and some cell type could be more prone to adaptation and to certain environments than others that are really maintaining throughout evolution. And so as an example, we look at the least conserved genes, so the genes that were the most divergent across cell type and across species. And we look at where are these genes expressed in our data set. And I just wanted to show a quick example. When we look at the most divergent genes in columella, what we found is that lots of these genes were expressed actually in maize. So here each line is a gene, you have like hundreds of them. And these genes are very poorly expressed in sorghum columella or in Ceteria columella. And when we look at geoterms in these genes, we found a lot of manosyntase, polysaccharides, biosynthesis, and lots of genes involved in mucilage biosynthesis or mucilage production. And so mucilage is an extremely important component of the root system in maize, and especially for microbiome interaction or nutrient. And so when we look at mucilage genes, so here's a just example of mucilage genes that have been the mostly characterized in Arabidopsis, for example. So here are the autologs. We found that these genes were highly expressed in the maize columella, but almost not expressed in sorghum except 2 autologs here. And interestingly, when you look at other cell type, and especially cortex, then it's the other way around, where here in maize cortex, you have very few expression of only a certain mucilage, but when you look in sorghum cortex, then all of these genes, except one here, are highly expressed in the cortex. So we think that there is a rerouting of mucilage production between maize and sorghum, and so we have multiple other genes that participate to the different component of the mucilage that still agree with this switch. And actually, in a quite old paper from 1970s, they were already like noticing that sorghum is able to secrete mucilage-like compound throughout epidermis at a higher level, and it wasn't possible in maize, where the mucilage was mostly localized in columella or produced by columella. So we currently working on that and try to prove that in different ways. And it's still a work in progress. So that's all for me. I wanted to thank the team, of course, for the amazing atmosphere we have in the lab. Of course, my PI, Kenneth Birnbaum, for his constant enthusiasm about science and all his support. I want to thank Ramin Rani for his help in the microscopic pictures and all his help in the root regeneration project, and all the rest of the lab and my support Human Frontiers grant. And so I also wanted to advertise, because we have a new postdoc position available in the lab starting in January. So if you know anyone, or if you yourself are interested to join the lab, feel free to reach out to me or to Ken directly by emails and tell us a bit about yourself. Thank you very much.